students. Today we are going to talk about isolation of plant genomic DNA and its purification, one of the important topic of practicals of genetic engineering and molecular biology students. The key topics being covered in this video are DNA extraction related equipment and necessaries, chemicals and buffers for plant DNA extraction, CTAP plant DNA extraction method, modification in CTAP DNA extraction protocol, and tips to care about. As we all know, DNA is a polynucleotide molecule which is made up of adenine, thymine, cytosine, and guanine with a backbone of mm, phosphate, and there are hydrogen bonds present between these nitrogenous bases. Extracting the DNA from the cell is nothing else, only the disrupting the cell wall by and cell membrane and nuclear membrane with the help of the chemical, physical, or mechanical method is basically known as DNA extraction. In 1980, Murray and Thomson developed an inexpensive, simple protocol for plant DNA extraction with the help of CTAB. Later, Taylor and Powell made it possible to extract high molecular weight, which is greater than 50 kilo base pair. Earlier, this CTAB method, cesium density gradient method, were used, and further Della Porta 1983, Shaghai and Maroof and co-workers in 1984 are the pioneers in developing plant DNA extraction method. Extraction of DNA from plant cell is little difficult as compared to animal cell, as plant cells are composed of cell wall, and which is a little bit tough to break this cellulosic cell walls. And also plants, they contain chloroplast, the photosynthetic organelle of plant cells, and this organelle, besides synthesizing of primary metabolites, is synthesize secondary metabolites like polyphenols and tannins, which mainly cause hindrance in extraction of DNA from plant cells. So it is a little bit more difficult to extract DNA from plant cells in compared to extracting DNA from animal cells. So, major steps in plant DNA extractions are lysis of cell wall with the help of physical methods and chemicals such as grinding and using liquid nitrogen, separating DNA by extraction buffer including CTAB and SDS-like chemicals, precipitation of DNA by alcohol, purification of extracted DNA by 70% alcohol, and dissolving DNA by trace EDTA buffer. Now we will talk about the equipments. The equipments regarding DNA extraction are very simple: weighing machine, autoclave, vortex mixture, centrifuge, pipettes and their autoclavable tips, liquid nitrogen container, ice box with ice, pre-chilled pestle mortar, spatula, different size of measuring cylinders, different size of flask, beakers, and appendrop tubes, which is the main part of DNA extraction buffer. Now, the chemicals which are majorly required in isolation of DNA are one of the important chemicals is liquid nitrogen. During the grinding, the heat is generated which activates enzymes such as endonucleases and exonucleases. These enzymes cleave our DNA into small pieces. Adding liquid nitrogen prevents the enzymatic reaction of these endonuclease and exonucleases. Liquid nitrogen, as we add the liquid nitrogen during the grinding of plant tissue, it immediately stop all the cellular reactions which are being carried out inside the plant cell, and by this we protect our DNA by harmful effect of DNAs and RNAs, and their degradation is being prevented, and it also helps in making homogeneous mixture of the tissue, which instantly froze at the point where it becomes ash. Now, the major, another major, we can say, chemical of DNA isolation from plant cell is the extraction buffer or lysis buffer, which is composed of mainly CTAP solution. Uh, and if we talk about the extraction buffer, the components are CTAP solution, NaCl, tris, EDTA, PVP, beta mercaptoethanol, and milky water, which is needed to make up the final volume. Here we have given two different methods to make this CTAP extraction buffer. One method in which we can prepare stock solution, and then desired volume can be taken and final volume can be made. Here we have given the calculation to make 30 ml extraction buffer. And another method is directly weighing of the chemicals and final makeup with the milky water. Here we are talking about the 30 ml final volume of CTAP buffer. So required concentration are fixed, like CTAP solution in the final extraction buffer, it should be 2%, NaCl 1.4 molar, So accordingly, we can make either stock solution or either we can directly weigh and make our lysis buffer 30%. Now, the important compounds which are being used in the extraction buffer, what are their roles? CTAB, which is commonly known as cetyl trimethyl ammonium bromide. It is one of the important compound. It acts as an cationic detergent, and it can bind with the cell wall polysaccharides by complexing with it. It co-precipitates the macromolecules along with the DNA, which is removed by the centrifugation. Letter note. So CTAB is the main compound which is used to complex the polar and non-polar compound present in the plant cell wall. Another important molecule is NaCl. The complexes which are formed by the C-tabs, they further being separate out by their differential solubilities in the presence of C-tab, which depends on the concentration of NaCl. So NaCl is one of the important factor, because at the low concentration DNA is insoluble, while polysaccharides are soluble at the low concentration of the NaCl. So accordingly, we have to add the concentration of NaCl. Now, third important component of extraction buffer is tris, which is basically the biological buffer, and its major role of tris is in any biological practice is to maintain the pH of the solution during cell lysis, removal of unwanted cellular components, and precipitation. Additionally, it plays a particular important role in cell lysis also, and furthermore, it also increases the permeability of cell wall. The fourth important compound is EDTA. EDTA majorly EDTA is a water and soluble organic molecule, and it can be dissolved in water at pH which is 8.0. EDTA is basically working here as gelating agent. As we break down the cell wall, cell membrane and nuclear membrane, DNA come out, and it can along with it, DNAs and RNAs enzymes become activated by reaction with their cofactor like magnesium and calcium ions. So EDTA, it easily bind with the calcium and magnesium ion and prevent them by adding with the DNAs and RNAs, and in the absence of magnesium and calcium ion, DNAs and RNAs, they cannot degrade our DNA, which is now directly in contact with these enzymes. So EDTA is one of the important factor which act as a gelating agent here. Another important compound which is being added in the extraction buffer is polyvinyl pyrrolidine (PVP). It basically binds with the phenolic ring of tannin and prevents their interaction with the DNA and protects it. So it is optional component. It depends on how much the secondary metabolites are present in our plants. Another important compound is beta mercaptoethanol. Beta mercaptoethanol, it also help in the removal of tannins and other polyphenols which are present in the crude extract. It may also help to denature protein by breaking disulfide bonds between cysteine residues. The storage of beta mercaptan is basically at 4 degree centigrade temperature. Now another important component is magnesium chloride. Magnesium chloride is also optional, we can add. It also protect our DNA from the action of the DNA's enzymes. Now another important enzyme is RNase A, which can be used for the removal of RNA for RNA-free DNA purification. Now another important, after extraction of the DNA from the plant cell and its removal of unwanted RNA and protein, the uh, supernatant is being treated with the chilled ethanol for the precipitation of DNA. As DNA is polar due to highly charged phosphate backbone, if enough ethanol is added, the electric attraction between phosphate group and any positive ions present in the solution becomes strong enough to form a stable ionic bonds, and DNA precipitation can be done. This usually happens when ethanol composes over 64% of the solution. So here chilled ethanol, it acts as an precipitating agent, and further 70% ethanol is being used for the purification step. 2-3 time wash with the 70% ethanol easily remove all unwanted compounds present in our DNA palette. So final the pellet is being stored in the TE buffer or milky water for its for prevention of DNA further degradation. One thing which should be remember, the autoclaving of all the solutions like CTAP solution, CTAP extraction buffer and SDS solution is recommended. Only the phenol and alcohols are not being autoclaved, along with different plasticares, glasswares like pestle mortar which are being used in the whole process. Tips, apentropes are already autoclaved, they can be used directly. So this should be done before doing this. Now the basic protocol for DNA isolation is, we will start with taking 50 milligram of fresh plant tissue, cut it into small pieces and grind it well. Then add liquid nitrogen carefully and again grind it again even harder until the tissue becomes powder. After it becomes homogeneous, take the mixture into 2 ml Eppendorf tube. Add 500 microliter of CTAP extraction buffer. Now mix well with the help of vortex mixture for 5 minutes. Incubate the sample for 30 minutes at 60 degrees centigrade to 65 degrees centigrade. Now centrifuge the sample at 10,000 rpm for 10 to 15 minutes and transfer the supernatant into another tube. Be careful, take only the supernatant, don't mess with the supernatant with the debris. Now add 5 to 10 microliter of RNA solution to the supernatant and incubate at 37 degrees centigrade for 25 minutes. Now add 5 microliter to 10 microliter of proteinase K solution to the supernatant and incubate at the 60 degrees centigrade for 2 hours. This step is additional. If it is required, then we can add the treatment of proteinase K. Now uh, centrifuge the sample at 10,000 rpm for 2 minutes and take supernatant to another tube. Now add chilled isopropanol. 70% of chilled isopropanol is added, which is 1.5 times more than the initial volume of the supernatant. Now precipitate the DNA by inverting the tube several times. After centrifugation at 10,000 rpm 10 minutes, throw the supernatant and now wash the DNA pellet with pre-chilled 70% alcohol 2 times at 9,000 rpm for 2 minutes. After the clear pellet appears, add 500 microliter of TE buffer or elution buffer to the pellet and dissolve the DNA. This is the diagrammatic representation of the complete process. Here we can see the complete process. Before step 6, we can add the PCI solution. It is additional step. Here P stands for the phenol, C for chloroform and I for isoamyl alcohol. Phenol, this is the ratio 25 is to 24 is to 1. If we are making 50 ml solution, then we can take this concentration of these 3 compounds. Phenol denature the protein and further addition of chloroform can enhance the complete denaturation of protein and lipid also. Here isoamyl alcohol can act as an anti-foaming agent, helping the clear separation of organic and aqueous phase. If we add the PCI, then we have to centrifuge the complete extract at 10,000 rpm for 10 minutes and then we will take the supernatant and here the debris will be the extra proteins and some other macromolecules. Most of the co-workers use this PCI solution. Here we can see that precipitated DNA can be obtained. If we can revise that cell extract after treating with the CTAP, it precipitated with nucleic acid and CTAP complex is formed. After centrifugation, nucleic acid CTAP it precipitated and its supernatant carbohydrate proteins has become aggregated, and after discarding it and resuspending in 1 mm NaCl and ethanol precipitation and ribonuclease treatment, we can get the pure DNA. It is another simple modification. The complete treatment depends on the requirement and sometimes on the trial. So modification in the protocol is again the depending on the plant material which we are being used. Either it contains high amount of secondary metabolites or polyphenols, so we can add accordingly different compounds, like we can use SDS, which is an anionic detergent. We can use urea at the initial state in place of CTAP. And finally the tips to in the complete process, we have to very much careful in using the different kind of compounds. We must wear the gloves and lab coat should be wore during the complete process, and milky or nucleus free water should be used for the preparation of chemicals. We should take care of tannins, which are the villain of the complete process. If the compound is rich in the polyphenols and secondary metabolites, then we should add the extra steps accordingly. And the storage of different compounds is different, so we should take care of that thing also. That's all about. Thank you so much. For further visualization